Good evening. I'm Sudiksha. Today we'll be discussing the contents of week 9, followed by some questions related to that. I'll just share my screen. If there are any doubts while I'm talking, please stop me and ask questions. One second. Okay. So, till now we have looked at classification, evolution, what are cells, what is the structure inside it, what are the biomolecules that make up these cells, and we have also looked at various cellular processes. Right now we are looking at physiology and what are the different systems that control the functioning of a normal body. So last week we looked at the digestive system, which is very essential for how we utilize the nutrients. This week we'll be looking at the circulatory system and the muscular system. Let's first look at circulatory system. The circulatory system is very essential for transporting gases, nutrients, waste, and hormones. Uh, if the body needs to exchange oxygen and carbon dioxide, it needs the circulatory system in place. It is also essential for transporting nutrients from where it is taken up, which is the alimentary canal, to different parts of the body where it can be utilized. The waste that are produced in different parts of the body uh, needs to go to the excretory system, and whatever hormones are produced in different glands need to different endocrine glands need to go to their respective places. The basic layout of a circulatory system is a pump, which is the heart in this case, the fluid which circulates through the system, and a series of pipes, which in this case is the blood vessels. There are two major types of circulatory system, one open system and a closed system. The open system is found more in lower vertebrates, wherein the fluid circulates into a chamber, and the exchange is mostly through diffusion, whereas in a closed system the fluid circulates through blood vessels. And in this system, the fluid needs to reach to each of the organs, and there is exchange of gases there. Open system, as I said, is usually found in case of lower vertebrates, especially in case of arthropods and mollusks, in which case there is a the blood in these animals, the blood adjacent is hemolymph, whereas the body cavity where the blood uh, is there a question? Whereas the body cavity where the hemolymph flows is the hemocoel. Uh, in case of arthropods like grasshoppers or cockroaches, there is a series of hearts that circulates the fluid. Each of these will have their own rhythm of sorts, and that's what enables exchange of different material. There will be certain openings that allows for the diffusion of the hemolymph into the outside of the hemocoel. Closed circulatory system is usually found in vertebrates, annelid worms, and some mollusks. In these cases, the blood moves through blood vessels by the heart's action. The heart has a certain rhythm to it. Its pumping action is what moves the blood through the whole body. And it does not come in direct contact with body organs. Rather, there is a layer of capillaries or smaller vessels between the organ and the blood. Uh, so there are different kinds even within the closed system there are different kinds of heart. The simplest one is a two-chambered heart, which is seen in fishes, wherein there is just one ventricle and one atrium. The atrium receives blood from the body. This is the deoxygenated blood. This, the blue one, is the deoxygenated blood. The ventricle then sends the deoxygenated blood to the gills, which is rich in capillaries. So there is exchange of there is CO2 that comes out and oxygen that goes in at these places. Therefore, the blood here becomes oxygenated. This oxygenated blood can then flow through the body, go to the capillaries present at different organs. There is uptake of carbon dioxide and uptake of oxygen by the organs, and release of carbon dioxide into the blood. And therefore, the blood then is deoxygenated, goes back into atrium, and the cycle repeats itself. Next is a three-chambered heart, where there is a separate atria that allows some separation of oxygenated and deoxygenated blood. So right now here at the body capillaries or at gill capillaries, there is not much of a distinction between deoxygenated and oxygenated blood. It's only the net difference that is going to lead to the exchange of carbon dioxide and oxygen. Whereas in case of a three-chambered heart, because there is a separate atria, so in this case there is a left atrium, there is a right atrium, but there is a single ventricle. This is primarily seen in land organisms such as reptiles and amphibians. There is some mixing of the blood in the ventricle, but the mixing is minimal, and even within this there are certain reptiles such as crocodiles which have a partial separation of the ventricle, so they essentially technically have a four-chambered heart. The complete four-chambered heart is seen in birds and mammals. There is a complete separation between oxygenated and deoxygenated blood. This is essential for temperature regulation especially, and for a fast metabolism. So this is found mostly in homeotherms or animals wherein they can control their temperature. The internal temperature of the body is not dependent on the external temperature of the environment. So in case of a four-chambered heart, there are two atriums, a left and a right. There are two ventricles. The blood flows from the left side to the lung, from the right side to the lung capillaries. Then the heart then receives the oxygenated blood from the lung, which then goes on into which is pumped into the body through different channels, finally goes into the organ systems, into the organs, where there is a thin layer of capillary which can allow for the exchange of blood, exchange of different nutrients and blood. So the four-chambered heart, because there is a complete separation, essentially has two pumps. There is the first one. In the first case, the atria contract, forcing blood into the ventricles. Is there a question? If there is no question, I would request that you shut your mic off because it's causing disturbances. Thanks. Okay. The first step is the atria contracts, forcing blood into the ventricles. The ventricles can then contract and force the blood through the arteries into lungs or to the rest of the body, depending on the site. And then the cycle ends, the heart relaxes. It allows for more blood to flow into the atrium. So the atria, as a rule of thumb, receive blood from the body or the lungs. Contractions of the atria send the blood through a valve into the ventricles. The ventricles receive blood from the atria and then send the blood to body or lungs. So atria receive blood, whereas ventricles send the blood. Depending on the site, the blood will be deoxygenated or oxygenated. Okay, so a human heart looks something like this. Aorta, superior vena cava, pulmonary artery, pulmonary veins, all of these are blood vessels, major blood vessels that originate from the heart and send blood to different portions. There is the right atrium here, there is a there is the left atrium here, left ventricle, right ventricle. Then there are different valves within the heart that prevent backflow of the blood. So the blood that comes from the atrium into the ventricle needs to stay in the ventricle. It shouldn't go back into the atrium. So there are unidirectional valves in place that make sure that the blood remains where it should be. If there are any defects in these valves, it can lead to mixing of deoxygenated and oxygenated blood, or it might need to lead to other heart problems. Some of these problems might be genetic. Some of these problems might occur in infants, wherein there is a thorough checkup done, and if there are any defects to any of the valves, the valve is repaired. Some of the valves are atrioventricular valve, semilunar valve, there is a mitral valve. Depends on and depending on how much pressure the valve needs to handle, it may be tricuspid or bicuspid. Okay. The rhythm of the heart is maintained by some nodes that are present within the heart. These nodes are can function independently, but there is also some amount of control of the nervous system on these nodes. There are two major nodes, the sinoatrial node or the SA node, that controls the primary beating of the heart. It causes the atria to contract and sends signal to the atrioventricular node to signal the ventricles to contract. So first the SA node should contract. Contraction, the signal at the SA node then needs to travel through the excitable fibers to the AV node. The AV node then makes sure that the ventricles are contracting to send the blood into different parts. Both of these nodes are also innervated by other nerves. For example, if you are exercising, or if there is release of adrenaline, both of these conditions leads to an increase in heart rate. So whenever such a situation arises, the nerves that are innervated here make sure that the SA node and the AV node get the required signals of sorts to make sure that the heartbeat rises as per the need. The blood vessels are the other component in the circulatory system. They fall in three major categories: arteries and arterioles, veins and venules, and capillaries. The arteries and arterioles carry blood away from the heart. These also usually arise from the atrium. The veins and venules carry blood to the heart. Arteries and arterioles arise from the ventricles, and veins and venules arise from the atrium, and they are spread throughout the body. At the organ systems, there are capillaries, which are one cell thick usually, which allow for nutrients, gases, and waste exchange. They also are very thoroughly spread, so that there is an increased area, and depending on the area, the exchange becomes better. So the arteries are thick walled, lined with smooth muscles. Therefore so the arteries, usually inside the artery, the blood flows with a very high pressure because the blood is pumped from the heart. So the arterial walls need to be thick enough to withstand that pressure. Once the arterioles are the just a branch off of the arteries, main arteries. These can constrict to direct and control the blood flow. They are slightly thinner than the arteries. They can increase or decrease the blood supply to the skin depending on the need. There is some control here. The body tissues contain a vast network of thin capillaries. They are only one cell thick, allowing for the exchange of gases, nutrients, and waste. They are also very thin that the RBCs must line up in a single file to go through them. So they are in the order of few microns. There are some spaces in between in case immune cells need to go from the bloodstream to into the organ system or into the lymph. Veins have thinner walls than arteries, so the blood from the arteries is coming from at a very high pressure. By the time it reaches the capillaries in the organ systems, it has lost most of its pressure. This is also very essential that the blood loses the pressure, otherwise it's going to damage the organ systems and the capillary network because they don't have much strength to them. By the time the deoxygenated blood enters the veins, it has very little pressure. Therefore, it does not need to have, the veins do not need to have very thick walls. However, they do need to have valves which prevents the backflow of blood. So at different places the veins have valves like this which allow for only unidirectional flow of blood. It does not allow for a bidirectional flow of blood. This is also very essential to make sure that the blood does not mix. Venules are thin-walled collectors of blood. The low pressure in the venules allows from the capillary beds to drain into them. So the pressure gradient is such a way, there is the artery, then there is capillary, then there is veins. Of course, the top has the heart. Okay. Okay. Next component in the circulatory system is the blood or the fluid that makes up the medium in the circulatory system. Within the blood there are different components. There is the plasma, which is the liquid portion. The plasma has different minerals, it has different proteins that needs to circulate freely. There is some amount of nutrients free floating within the plasma. So essentially the plasma functions as the medium to deliver everything. Then there is red blood cells, which is the major portion of the blood. We will be discussing some of it in some details in the coming slides. There are the white blood cells which protect the human organism from different infections. Then there is platelets. If there is an injury at any site, the platelets make sure that there is not a lot of blood loss. RBCs, so the RBCs that humans have do not have any nucleus. Initially, when the RBCs are produced in the bone marrow, they have a nucleus, but they lose their nucleus at maturity. There are certain this is true for most mammals, but there are certain exceptions to it. For example, camels don't lose their nucleus. Their RBCs still have the nucleus. RBCs are usually concave in nature. They make up about 99% of the blood cellular component. RBCs in general have a red color due to the presence of hemoglobin. The hemoglobin is a complex protein made up of 4 protein strands and a heme group, which is attached to an iron. So there are 4 polypeptide chains folded, and each of these have a heme group inside them. The heme group can bind to the uh, iron Fe3 plus ions. These molecules can, each hemoglobin molecule can carry 4 oxygen atoms attached to each of the heme groups, and if there is oxygen present, the hemoglobin is bright red, otherwise it's more bluish. So arterial blood is bright red in a color, whereas venous blood is slightly darker in color. If the arterial blood is also darker in color, then there is some issues with the iron carbon dioxide system. RBCs live for about 4 months, and when they die, they go into liver to get recycled by liver and spleen to get recycled. The iron from the hemoglobin is taken out and can be used again for making new RBCs. The hormone erythropoietin which is made by kidneys, is the one that stimulates the production of RBCs in the red bone marrow. If there is an oxygen deficiency, this stimulates erythropoietin production by the kidneys. Once erythropoietin is produced, it stimulates the bone marrow to produce more red blood cells. This causes the oxygen deficiency to go down and the oxygen level to increase. Restored oxygen level then inhibits erythropoietin production by the kidneys. If there is an oxygen deficiency again, remember this, you need to remember that this oxygen deficiency is slightly chronic in nature. If for example someone is exercising for like 5 minutes, if there is some deficiency, it might start the cascade, but the deficiency is not enough that the whole cascade goes through. It's only if there is a prolonged deficiency, only then the cascade starts. Okay. Next are the white blood cells, which defend against diseases by recognizing proteins that do not belong to the body. These can go out of the capillaries to patrol different tissues. If there are any infections within the tissues, they activate themselves, and then they can clear out the infection. Okay. The white blood cells form most of the immune system, which protects organisms against invading pathogens. These are usually specific or non-specific reactivity induced by a host in response to something that is not present in, that is not self. So the immune system has the ability to distinguish between self and non-self. If the immune system finds something that does not have the markers of self, then it knows that these cells are not from my own body, therefore I need to mount an immune response against it. The self and non-self distinctions comes from antigens, and because of the presence of the antigens, the immune system mounts an immune response. The immune response can be of 2 types, humoral mediated immunity or cell mediated immunity. Humoral mediated immunity comes from the plasma portion of the blood, whereas cell mediated immunity comes from the cellular portion. The cells release different antibodies, complements, other components that are essential for immunity, and this is what is always flowing through in the body. This is especially important for defense against bacterial pathogens and viruses. Cell-mediated immunity involves cells such as T cells, B cells, essentially various lymphocytes. They protect against fungi, virus, and various bacterial pathogens. The immune system also provides some protection against cancer. Is there a question? Okay. Okay. And if there a lot of tumor suppressor, a lot of tumor evasive pathways also depend on the fact that cancer cells have certain cancer cells have the ability to sort of cheat the immune system into thinking that they are normal cells. Immune response is a multi-step process. When the pathogen enters or the antigen enters, the macrophage takes up the antigen, then does processing, then presents it with MHC class 2. MHC2 along with the antigen can then bind to helper T cells. This activated helper T cells, because they now know what the antigen is, they can then activate B cells. The activation can be of two types. Either the B cells can form plasma cells, which will produce antibodies, or some of the B cells, which know what the antigen looks like, form memory cells. These memory cells are essential for the immune memory. If there is another infection from the same pathogen a second time, then the memory cells recognize that, yeah, we have seen this antigen at some point and we know how to attack it. So therefore these memory cells can signal formation of other plasmas, of new plasma cells, without going through a lot of processing. The antibody that the B cells produce looks like a Y-shaped object. There are light chains and there are heavy chains. The heavy chains make sure there is also a constant portion, which is known as the effector region, and there is a variable portion. The variable portion, because the sequence and therefore the binding site in the variable portion is going to change with each antibody. Because of this, it makes sure that there is a large variety of antibody in place, and one or the other antibody can bind to the antigen. The constant region is the effector region. It has the signaling in place for it to if there is a antigen bound to the variable region, then the effector region can then signal for the downstream processing. So the effector region is more or less constant. The variable region is the one that changes and molds itself according to the antigen. Production of antibody occurs in four stages. There is a lag phase where there is entry of pathogen and detection by immune cells. There is a log phase where there is a steady increase in the titer of the antibody. There is a plateau phase where there is equilibrium between antibody synthesis and catabolism or breakdown. At this point, during the plateau phase, more or less the infection has been cleared out. Once the infection has been completely cleared out, there is a decline phase wherein the antibody amount goes down, or the titer goes down, because the body no longer needs the antibody in place. Everything that the body synthesizes has a cost associated with it. So if the body does not need a particular antibody, it do not need to synthesize it. So next are the platelets, which are cell fragments used in blood clotting. They are derived from megakaryocytes, and like RBCs, they also lack a nucleus. They have a very short lifespan, usually about 10 days, and therefore there is always a constant population of platelets, but they are dynamic in nature. Some of them get removed and new ones get added. They aggregate at the site of the wound. So if there is a break at any point in the capillary, the injury signals platelets to come and start binding here. Uh, Usually, the capillary bed is covered by different components that make sure that the platelets remain in circulation, they don't stick there. If there is an injury, the extracellular matrix proteins, which are the proteins present outside of the cell, which make the meshwork that make sure that the cells don't move around, is damaged. And most of the extracellular matrix is made up of collagen fibrils. These collagen fibrils, the platelets can bind to these collagen fibrils. So in a normal state, because the collagen fibrils are not exposed, the platelets won't aggregate. Whereas a case where there is an injury, the platelets start aggregating because the collagen fibers are exposed now. Then they start releasing chemicals, they stimulate thrombin production. This thrombin converts one of the proteins known as fibrinogen to a sticky fibrin which binds the clot. So it makes a mesh of fibrin plus plus the platelets to make sure that there is no leakage of the blood to the to outside of the blood vessel system. Next, as I said, the blood within the arteries flows through very high pressure. This leads to the phenomenon of blood pressure, wherein which is of two types. There is systolic pressure or diastolic pressure. This dictates what kind of flow rates go through in the blood vessels. The systolic pressure is the pressure when the heart contracts, and diastolic pressure is the low pressure when in between heartbeats. So when there is contraction, the blood is flowing at a very high rate. So the normal value for that is around 120 mm of mercury. Diastolic pressure is, since it's between heartbeats, it's not at as high as high pressure, it's around 80. If the values of either of these go up, consistently stay up, it's called hypertension. Whereas if the values consistently stay down, then it's known as hypotension. Hypertension puts high blood pressure puts people at risk for heart disease. If the blood is constantly circulating at a very high pressure than what the body is used to, then it will start damaging damaging the arterial walls, it will damage the capillaries, it will damage different organ systems that are involved. Another problem that occurs in the circulatory system is atherosclerosis, where there is a plaque formation. This plaque is usually made up of cholesterol. So what happened? Cholesterol is of two types: LDL, or low density lipoprotein, or high density lipoprotein. Sorry, lipids are of two types: low density or high. Lipoproteins are of two types: low density or high density. If the body is rich in low density lipoprotein, that implies the signals in place make sure that the cholesterol comes into the circulation. Whereas if there is a high amount of high density lipoprotein, the signaling is such that the cholesterol stays in storage and does not circulate. Therefore, when there is very high LDL, cholesterol is in circulation, and this cholesterol forms plaques in arteries. These plaques trigger inflammation. There is rupture of the if there is too much inflammation, then there is rupture of the plaque that is formed, and that mounts an immune response. The immune system forms a hard cap over the plaque, partially blocking the artery. When the caps rupture, create clots that can close off arteries. So this has multiple problems. The first problem is inflammation. The second is if in case, because there is constant blood flow, in case there are the caps get ruptured, then it can form clots. These clots can then go and lodge at different places within the artery, and this might create blockages. Uh, these blockages can then finally can also result in heart attacks. Heart attacks can come out of genetic and environmental factors. Both of these also trigger atherosclerosis. Uh, LDL can be reduced by low fat diet, which emphasizes on high fiber food, antioxidants, good fats, which are usually monounsaturated fats, omega 3 oils, and reduced trans fats. So in general, saturated fats come in LDL, whereas monounsaturated fats come in the HDL fraction. Regular exercise also contributes significantly to LDL cholesterol reduction. Okay. The next, just give me a moment. Are there any questions till now? Okay. Let's discuss muscular system then. Musculoskeletal system. The muscular system is very essential for locomotion, which is the ability of organisms to move from one place to another. There are various organs that help with locomotion, and these are known as locomotive organs. These will be different for different organisms. For example, it may be limbs. For unicellular organisms, it may be flagella or cilia. The muscular system is a type of tissue among 4 different tissues. They are a specialized type of tissue, and they originate from mesoderm. So when an organism is developing, a triploblastic organism is developing, there are 3 layers to it. During development, there is the endoderm, which gives rise to certain organs, mostly the alimentary canal, various all of the alimentary canal is derived from the endoderm. There is the mesoderm, which gives rise to muscles. There is the ectoderm, which gives rise to skin, nervous system, etc. Muscles alone contribute to 40 to 50% weight of the human. The characteristics of muscle cells that make sure that they do the function that they are doing are excitability, contractility, elasticity, and extensibility. So muscle fibers are excitable, they have the ability to contract, they are elastic nature, so there is some leeway to how much they can stretch, and they can extend. Based on the location, muscles are classified into 3 categories: skeletal muscles, visceral muscles, and cardiac muscles. Skeletal muscles are associated with the skeleton, the skeletal components, essentially the bones. They look striped in nature. They are also known as striated muscles. Most of the skeletal muscles are regulated by voluntary movements by the nervous system. It's only if you want to move that muscle, only then you move it. The principal action of skeletal muscle is locomotion and modulation of the body shape. So, for example, if you want to pick up something from the table, the muscles that play a role in this is the skeletal muscle. Next are the visceral muscles. They are present in the inner walls of visceral organs of the body. Example, alimentary canal, reproductive tract, etc. They are not voluntary, they are involuntary in nature, and they are mostly responsible for transport of various things within the body. Next are the cardiac muscles, which make up the heart. These cells are assembled in a branching pattern. So cardiac muscles look a lot like the skeletal muscles, but the only difference is they are branched in nature. They are also involuntary. So cardiac muscles come somewhere in between. They have some function, some properties of the skeletal muscles, and some of the visceral muscles. So major differences: skeletal muscles are around the bone all over the body, whereas visceral muscles are form the inner walls of hollow organs, and cardiac muscles form the heart. The morphological appearance in case of skeletal muscle is striped, visceral muscle is smooth, and in case of cardiac muscle it's striped with cross-linking. Except for skeletal muscles, both of the other muscle types are involuntary in nature. Skeletal muscle is voluntary in nature. Major functions of skeletal muscle is movement, for visceral muscle is transport of different nutrients or cells. Cardiac muscle is pumping. There are different contraction types. In case of skeletal muscle, it's short twitch and long tetanic. Not tetranic, tetanic. In visceral muscle, it's concentric, eccentric, isometric, or isotonic. And in case of cardiac muscles, it's long tetanic. Skeletal muscles are elastic in nature. All of these muscles are elastic in nature. Sizes of different muscles also vary depending on the location. Skeletal muscles can range from 10 to 80 micrometer. Visceral muscles are 1 to 5 micrometer, and cardiac muscles is around 10 micrometer. So skeletal muscles are the cells that make up skeletal muscles are also known as muscle fibers. They're essentially long cylindrical multinucleated cells, so they form something known as a syncytium. Since a syncytium forms where the cytoplasm of different cells are fused, and the cells have, so essentially there is one big cell which has a lot of nucleus. Muscle fibers are organized at 3 levels: epimysium, endomysium, and perimysium. Muscles are covered by thick, tough connective tissues known as epimysium, which forms a big bundle of muscle fibers. Beneath the epimysium there is the perimysium, which covers 100 to 150 muscle fibers and form fascicles. Fascicles are essentially covered by perimysium. The interstitial space between muscle fibers is around 1 micrometer, which allows for development of tunnel in the perimysium, which is the place where arteries, veins, and nerves pass through. The collagen fibers are arranged in wavy forms with crosslinks that helps improve strength and stability of the muscle fibers. Endomysium consists of loose connective tissues, and they additionally add up the strength of the muscle fibers. The endomysium also connects to the perimysium for stability. The basic structure that makes up a muscle fiber is the sarcomere, which is a multinucleated cell. These muscle fibers are lined by plasma membrane, which in case of skeletal muscles is known as sarcolemma. The sarcolemma encloses the sarcoplasm, which is like cytoplasm. As I said, muscle fibers contain multiple nucleus, which makes up the syncytium, and the sarcoplasm contains relatively high amounts of glycogen granules and myoglobin. The myoglobin are similar to hemoglobin in the sense that they are also used to store oxygen, but the binding affinity of myoglobin and hemoglobin to oxygen differ very significantly. A high amount of calcium is stored in the endoplasmic reticulum. In case of skeletal muscles, the endoplasmic reticulum is known as sarcoplasmic reticulum. Parallel arrangement of myofilaments in the sarcoplasm is one of the characteristic nature of muscle fibers. Two types of myofilaments known as myofibrils are present in muscle fibers based on the thickness. Myofibril are termed as thick or thin myofilament, which is made up of actin and myosin filaments respectively. So the thin filament is the actin portion of the sarcomere, whereas the thick filament is the myosin portion. There are because the actin and myosin filaments are arranged in a certain way, there are different uh, patterns that you see in sarcomeres. We will discuss this but in a bit. So in general there are different bands: A band, I band, Z zone, Z line, M line, etc., depending on what is present in that area. In general, the portion between two consecutive A bands is known as a sarcomere, which is the functional unit and is responsible for muscle contraction. There are different cells that make up the sarcomere. For the first major there are different proteins that make up the sarcomere. The first major protein is actin. It is polymeric in nature. So the actin monomers are known as G-actin or globular actin. Its molecular weight is around 43 kDa. A number of actin monomer units polymerize and form fibrous F-actin or filamentous actin. Two filamentous actin coil around each other to form one alpha helix, and the thickness of the F-actin filament is around 6 to 7 nanometer. It consists of 14 G-actin molecules per turn, and depending on how many turns are there, the number of G-actin molecules in filament will vary. The F-actin is associated with tropomyosin troponin. It forms the I band of the myofibril. Troponin masks the active binding sites for myosin on the actin filaments. So, on the actin filament, troponin sits at the myosin binding site. When the stimulus is enough, then the troponin leaves the binding site so that the actin can bind to the myosin. Myosin is another major muscle protein. It's hexameric in nature. There are 2 heavy chains and 4 light chains. The heavy chains are around 200 kDa and the light chains are around 15 to 27 kDa. Kilodalton, I don't know if you know this, but kilodalton is the unit of measure for size in proteins. And when you run a Western for these proteins, you would find denatured proteins at this size, essentially. The size depends on what is the amino acid structure of the protein. The heavy chain forms dimeric filaments by coiling alpha helically with each other except for the N terminal. The unwound N terminal of the myosin is globular in nature and serves as binding sites for other light chains as well as F-actin. So the globular head of the myosin is where the F-actin binds to. N terminal of the myosin has ATPase activity, and the carboxy terminal of myosin filament meet together at H zone, whereas N terminals meet together at the margins of the A band. Tropomyosin is a rod-like fibrous molecule. It has a molecular weight of around 66 kDa. It is made up of 2 different alpha and beta peptides, and both are alpha helical in nature. They are double-stranded and run parallel with the thin filaments in the grooves of F-actin strands. Troponin is a globular protein. It is made up of a 3 protein system: TPT or tropomyosin binding protein, TPC or calcium binding protein, and TPI or actin and TPC binding protein. The troponin lies on thin filaments at an interval of 38.5 nanometer. but the rest is attached to effectin and tropomyosin. The polymerization on this we will discuss later. So, as I said, the skeletal muscles are voluntary in nature. That implies there is a need for the signal to reach the skeletal muscle, uh, and therefore it is under the control of the nervous system. So the basic unit that controls what when the skeletal muscles are excited is the neuromuscular junction. So it is a junction between axon terminal of the neuron and the sarcoplasm. So the neural muscular junction is a connective region of a efferent nerve fiber of the nervous system and muscle fiber of the muscle system through synapses. So efferent means it is coming towards the muscle. The entire junction is covered by a cell which is known as a Schwann cell for insulation purposes here. Invaginated membrane appearance is present in this junction known as synaptic gutter or synaptic cleft, the neuronal end. Some neuronal clefts surround the axon terminal and it increases the surface area for synaptic transmitters. So in general, when there is a need for a movement, the synaptic vesicles here gets released into the synaptic cleft, and then they are detected by the sarcoplasm, which allows for the release of calcium ions which is present in the sarcoplasmic reticulum into the sarcoplasm of the sarcomere. This then allows for the movement of the muscles, the contraction within the muscles. Next is the visceral muscle, which are the smooth muscles. These are made up of smooth muscle cells which are fusiform in shape. They are sort of like spindle shape, but they can take up any shape that they want to. They are mostly present free in nature as in a single fiber form. It has a greater elasticity than muscle fibers. The contractility and extensibility is also higher than muscle fibers. Some of the proteins that are present are myosin, actin, troponin, tropomyosin, calponin, chyldosporin. Depending on what the function is, some or all of these proteins might be present. The relaxed smooth muscle cells look something like this, whereas contracted smooth muscle cells look like this. In case of smooth muscle cells, the branches of autonomous nerve fibers innervate the smooth muscles in a diffuse manner. These junctions are not connected directly with the muscles, but they secrete the neurotransmitters at the external space of the muscle fiber, and these diffuse through the muscle fiber membrane to transmit the signal, somewhat similar to the neuromuscular junction in case of skeletal muscles. Cardiac muscles have many features like skeletal muscles. They are striated in nature. They have a similar banding pattern, but in case of cardiac muscles, they are separated by dark areas which are known as intercalated discs. They are connected to each other. So in case of as a rough view, sarcomere will look something like this, whereas cardiac muscle fiber, because of connections, will look something like this. Interconnected cardiac cells are called as syncytium. Heart consists of two syncytiums, arterial and ventricular. Okay. So let me just pull up this portion. So there is as I said, there is a specific banding pattern for the skeletal muscles which enable it to do the locomotion. There are two different types of contractions that undergo in the skeletal muscle: twitch contraction and tetanic contraction. There are also other types of contractions such as concentric, eccentric, isometric, and isotonic. Cardiac muscle contraction is also a kind of tetanic contraction, but there are molecular differences between voluntary and cardiac muscle contraction. The skeletal muscles look something like this. So this is a eye band which is rich in actin molecules only. This is the M line or the middle line. There is a H zone which does not which is rich in myosin but no actin, and there is a Z disc wherein the actin and myosin filaments are connected, and there are these regions wherein the actin can bind to the myosin molecules. There is a Z cap which makes sure that the actin is stable, does not depolymerize. If there is a contraction, the distance between two Z discs decreases, or the distance between M line and Z disc decreases. Twitch contraction is a very short timed muscle contraction. It is usually in response to a stimuli in which muscles contract and relax, but before reaching the peak of contraction, the muscles start relaxing. Tetanic contraction is longer in nature. When there is a stimulus, the muscles contract, stabilize, and then relax in a sequence manner, and the duration of the stable contraction depends on the strength of the stimuli. Concentric contraction occurs when there is a need to overcome a resistance with some generated energy. The muscle contracts and shortens. This contraction can be seen in biceps during when lifting something or during angle change in the joints. Eccentric contractions occur when there is a lack of energy but there is an external response. In response to this external stimulus, the muscles contract and lengthen instead of shortening. The response is slightly delayed as compared to the concentric contraction because it's not a very active process. Isometric contraction occurs when there is generation of force but there is no change in the muscle length. Because of this, there is no movement. Isotonic contraction happens when there is excessive force on muscle, because of which it initially contracts and later there is an increase in the muscle length. The muscle length increase is in response to the external force. All of these contractions occur when there is an external stimulus. In general, if there is a stimulus, this signals for a generation of action potential from there are signals from the brain to generate an action potential. At the end of the axonic terminal, there is a small amount of neurotransmitter called as acetylcholine secreted. The acetylcholine diffuses from the membrane receptors which is present in the muscle membrane to transmit the signal. Acetylcholine binding leads to opening of sodium channels, resulting in the influx of sodium inside the cell. The sodium enters the cell, potassium ions go out of the cell. This generates an action potential in the muscle fibers and it travels along the muscle cell fiber. Stimulus by acetylcholine at one particular point triggers the generation of action potential in the nearby muscle fibers as well. This action potential depolarizes the muscle membrane, which leads to release of calcium ions from the sarcoplasmic reticulum, the sarcoplasma, to the sarcoplasma. Then there is calcium mediated attraction between actin and myosin, leading to sliding alongside with each other. Once acetylcholine is present, calcium is released. The calcium makes sure that the troponin which was present on the actin filaments gets released. This exposes the myosin binding site that is present on actin. So therefore the actin filaments can now bind to the myosin filament. Then there is a utilization of the of ATP, which gets released as ADP and PI. This release of the ADP and PI creates a sliding movement which results in the muscle contraction. After a fraction of a second, the calcium pump actively transfers the calcium from the sarcoplasm to the sarcoplasmic reticulum. And this removal of calcium from the sarcoplasm results in muscle relaxation. So this sliding movement that occurs between actin and myosin filaments is known as a power stroke. Multiple such power strokes might happen, and this leads to a more efficient contraction of the muscle. Muscle contraction mechanism is described by a sliding filament theory, which was described by Andrew Huxley, Rolf Niederkirk, Hugh Huxley, and Gene Hansen in 1954. Muscle contraction is a cyclic repetitive process in which actin filaments slide over myosin and generate tension in the muscle. So firstly, muscle is in the resting state. When there is a stimulus, calcium ions get released. They bind to the tropomyosin troponin. Then they make sure that the troponin is moved out of the binding site. Actin binds to the myosin. There is ATP lysis into ADP and PI. This actomyosin binding leads to contraction of actin filament. Then there is ATP binding again to the myosin, which releases the actin filament from the myosin filament. The released calcium ions from the tropomyosin then move back to the sarcoplasmic reticulum. Action potential from the central nervous system reaches the neuromuscular junction and release the acetylcholine near the muscle fibers. The acetylcholine diffuses the synapse where it diffuses through the synapse where it binds to the nicotinic acetylcholine receptors. After binding, receptors get activated on the neuromuscular junction and it leads to opening of sodium or potassium channels. This results in sodium influx and potassium outflux from the cell. The sodium potassium movement from the muscle cells generate an action potential which leads to depolarization of the inner muscle fiber. Once there is depolarization of inner muscle fiber, this activates L-type voltage dependent calcium channels. One such example is DHP or dihydropyridine receptors in the sarcoplasmic reticulum. These channels resemble the calcium release channels such as ryanodine receptors. Once these are activated, the sarcoplasmic reticulum starts to release the calcium channels. The released calcium channel binds to troponin C, which is present in myosin fibril, modulates the tropomyosin allosterically. So there is a structural change in tropomyosin. In normal condition, tropomyosin does not allow myosin to bind to the thin fibers or the actin fibers. But in presence of calcium, troponin undergoes allosteric modification and troponin loses the affinity over the thin filament. It results in tropomyosin migration from the thin filament, and immediately myosin binds strongly with the freed area in the thin filament. So there are binding sites between binding sites on the actin filaments and on the myosin filaments. These binding sites are masked because of troponin and tropomyosin. When there is calcium released, these sites get exposed and therefore actin can now bind to the myosin filaments. This process is an energy coupled process. So the myosin utilizes ATP energy by cleaving into ADP and inorganic phosphate. Actin, which is involved in this process, it helps releasing the inorganic phosphates from myosin, because actin has some ATPase activity in it. The overall result is sarcomere shortening, which implies that the distance between Z band shortens. The movement of the myosin was observed for around 10 to 12 nanometer per power stroke. Multiple such power stroke cycles can happen, and this depends on the availability of ATP. Um, the molecular mechanism behind it is something that we have kind of already discussed. In case of smooth muscle fibers, the troponin is replaced by P light chain protein. The instead of sarcoplasm, it's no longer sarcoplasm, it's cytoplasm again. Calmodulin and myosin light chain kinases are present. The action potential generated condition, the sarcoplasmic calcium level increases with calmodulin, which immediately binds with 4 calcium molecules. Calcium calmodulin complex immediately binds to the enzyme P light chain kinase. P light chain kinase calmodulin complex actively phosphorylate the P light chain, and the phosphorylated P light chain can readily dismantle the actin filament. And if the myosin binds with the actin after ATP lysis, there is a contraction of the actin filament. What you also need to realize is that the actin and myosin filaments are not static within any of these cells. They are undergoing dynamic remodeling as the power stroke is happening. If there are damaged actin molecules, or if some of the actin molecules need to be replaced, the replacement is also going on. And yeah, so when ATP binds with myosin, it again disassembles from the actin filament, and P light chain binds back to the actin to lead to a more relaxed condition. What are some of the muscular system disorders that happen? First is muscle strain, which is an injury in which muscle fibers tear as a result of overstretching. It's also known as pulled or torn muscles. Some muscle strain is okay, especially if there is exercise, if there is a very high intensity exercise going on. But a lot of strains might be a signal for some underlying cause. Hamstring strains are prevalent in track and field athletes. In sprinters, for example, 1/3 of injuries are hamstring injuries. Hamstring is a muscle within the leg. Having a previous hamstring injury puts an athlete at an increased risk of having another one. If the strains are very common, then, or if the injuries are very common, then someone needs to get checked for other underlying problems. There might be a possibility that the injury is not getting repaired properly. Tendinitis is an inflammation of the tendon that occurs when it's overextended or worked too hard without rest. The areas commonly affected are ankle, knee, shoulder, elbow, so joints essentially. Next is the carpal tunnel syndrome, which is a biomechanical problem in the wrist wherein the median nerve becomes compressed between the carpal bones, because of which there is a lot of pain. This may occur due to repetitive use of the wrist, a tumor, or a trauma to the wrist. Then there is muscular dystrophy, which is a genetic disorder caused by defective proteins in the muscle cells. It is characterized by progressive skeletal muscle weakness and death of the muscle cells and tissues. So usually this happens when improperly formed or misfolded proteins accumulate within the muscle cells. Myasthenia gravis is an autoimmune disorder in which circulating antibodies block nicotinic acetylcholine receptors on the neuromuscular junction. This blockage of the receptors causes muscle weakness, first exhibited by drooping eyelids and expanding to include overall muscle weakness and fatigue. Parkinson's disease is another autoimmune disease. It's a degenerative disorder of the central nervous system that mainly affects muscular system and movement. 4 motor signs and symptoms are considered defining Parkinson's disease: if there is muscle tremor or shaking, muscle rigidity, slowness of movement, postural instability. So in general, if the immune system fails to recognize, fails to differentiate between self and non-self cells, if it start recognizing the cells of the host system as an foreign body, then it starts mounting an immune response against it. This is when autoimmune disorders happen. Usually there are mechanisms in place that tell the immune system, yes, this is something that belongs to this individual, this is not a foreign object. But if these molecules that make up the identity of self are damaged or not produced properly, then there might be an autoimmune disorder. Are there any questions till now? Okay. If not, if there are no questions, then I will discuss some questions. If there are any doubts, please stop me. Okay. The first question is, the effect of increased calcium ion concentration on muscle contraction is it inhibits contraction by blocking actin sites. It triggers the exposure of actin binding sites for myosin. It stops the release of acetylcholine, or it relaxes the muscle fibers. Someone wants to answer? The answer is B. It triggers the exposure of actin binding sites for myosin. If there are calcium ions, it will make sure that troponin, tropomyosin interactions with the actin and myosin filaments break and the sites are exposed. The following is responsible for relaxation of muscle after contraction: release of acetylcholine, depletion of ATP, hyperpolarization of muscle cell, active transport of calcium ions back to the sarcoplasmic reticulum. The answer is active transport of calcium ions back to the sarcoplasmic reticulum. If the calcium ions is no longer present, troponin and tropomyosin can go sit in their respective places. The term for muscle contraction that generates force but does not change the muscle length is isotonic contraction, isometric contraction, concentric contraction, or eccentric contraction. Anyone wants to answer this? The answer is isometric contraction. So metric pertains to the length of the contraction. The role of calcium ions during muscle contraction is A, to trigger the release of ATP, B, to directly contract the muscle fibers, C, to bind troponin and expose binding sites on actin, or D, to directly contract the muscle fibers. The answer is C, to bind with troponin and expose binding sites on actin. During muscle contraction, the ATP provides energy to bind calcium to troponin, energy to open sodium channels, energy to transport oxygen to muscles, or energy to detach the myosin head. The answer is D, energy to detach the myosin head. Protein in muscle fibers that binds to calcium ions to initiate contraction is troponin, tropomyosin, actin, or myosin. The answer is troponin. Function of sarcoplasmic reticulum in muscle cells is transporting calcium ions to the nucleus, storing calcium ions for muscle contraction, producing ATP for energy, or synthesizing proteins. The answer is B, storing calcium ions for muscle contraction. The type of blood vessel that is responsible for nutrient and gaseous exchange is arteries, veins, capillaries, lymphatic vessels. Anyone wants to answer this? The answer is capillaries. The part of heart that pumps blood to the lungs is left ventricle, left atrium, or right atrium. The answer is right ventricle. The chamber of the heart that receives deoxygenated blood from the body is right atrium, right ventricle, left ventricle. The answer is right atrium. So the blood comes in from the body into the right atrium, then goes into the right ventricle. The right ventricle then pushes the blood into the lungs for get oxygenated. The oxygenated blood then comes into the left atrium. Then from the left atrium it comes into the left ventricle. From the left ventricle it can then send the blood to the rest of the body and to the muscles of the heart as well. The role of sinoatrial or SA node in the heart is pumping blood into lungs, maintaining blood pressure, secreting hormones, controlling the heart rate by generating electrical impulses. The answer is D, controlling the heart rate by generating electrical impulses. A 12-year-old accidentally cut off his finger while chopping vegetables. After a while, he noticed that the bleeding had stopped and a scab had formed. The component of blood that plays the most critical role in stopping the bleeding is red blood cells, white blood cells, platelets, or plasma. The answer is C. Sorry. The answer is C, platelets. Then they can signal other components from the plasma as well to come and staunch the bleeding. While digesting the food, stomach and intestines perform rhythmic contractions to move the food along the digestive tract, which is known as peristalsis. The feature of the muscle type that is responsible for this action that is making it suitable for such a process is involuntary control, voluntary control, striated appearance, or multinucleated. The answer is involuntary control. Otherwise, you would constantly need to think about food getting digested so that the stomach and intestines keep performing their function. If you want to pick up a book from a table, your brain sends a signal to your hand to perform the action, at which specific site does the motor neuron communicate with the muscle fiber to initiate this movement. Neuronal junctions, occluding junctions, plasmodesmata, or neuromuscular junctions. The answer is D, neuromuscular junctions. One more thing is motor neurons are the ones that originate from the central nervous system and innervate the muscle fibers. There are other neurons that innervate other places, but muscle fibers in specific are innervated by motor neurons. Basic contractile unit of skeletal muscle is sarcomere, myofibril, muscle fiber, or myosin. The answer is A, sarcomere. Okay. Are there any questions from any of the parts, either from the theory part or from the questions? I am here for a few minutes if someone wants to ask something. Okay. If there are no more questions, are there any questions? Okay. Then thank you for listening. This is the end of today's class. Thank you for attending.